So I think we should start the live session. So I'll share my screen. Just somebody let me know whether whether the slides are visible and I'm clearly audible or not. Okay, so hopefully I am clearly audible then. Fine. Okay, so again, I welcome you all to week 3 of the course Bioinformatics Algorithms and Applications, the course instructed by Professor Michael Gromiha from IIT Madras. And again, today's tutorial session I will be taking. So I am Sunanda Samanto, a PhD scholar from IIT Indore at the Department of Biosciences and Biomedical Engineering. Right. So the last time in the TA session, we covered some of the topics that relates to sequence alignments, right, on a broad scale. So can somebody remind me which topics we covered in the last class, in week 2's live tutorial session? So we covered some basic alignment visualizations, right? For example, in dot matrix. And then we gradually went over to some more, rigid kind of quantitative approach where we introduce some sort of scoring systems to actually quantify the alignment scores, right? To find out which alignment would be better in a way, right? So in this tutorial, we'll again go through some of that in the very beginning, and then we'll gradually move to some of the most useful or widely used algorithms that are used in case of sequence alignments, right? Okay. So does somebody remember what was dot plots? So what does a dot plot represent? How do we construct a dot plot? And what kind of pattern are we looking for when we uh, construct a dot plot using two sequences, whether that be protein sequences or nucleic acid sequences? You can unmute or you can write in the chat. The dot plots are basically a visualization sort of tool, right? When on both of the axes, right, on the x-axis and the y-axis, we just write down the sequences character by character, and whenever we find a matching character, we just put dots in there, right? So the entire basically matrix will be covered with dots. And what we are looking for in this particular dot plots, what we are looking for are stretches of diagonal runs, essentially, which will mean that those diagonal runs, if we kind of sort of go to the sequence 1 as well as to sequence 2, so we sort of find out which areas are making those diagonal runs, we can identify which portion of the sequences are aligning with one another, right? So that is sort of a visual approach to dot plot. And if we kind of go over the process again, so in this grid, I have put two sequences, right? One is ACGCG and another one is ACACG. And what we have to do is that if we start matching character by character, so you have to put dots in the particular places or in the particular junction points where the characters will be matching between these two sequences. So can somebody tell me whether we should put a dot in this particular junction point where the cursor is for a dot plot? If we, let's say, match character by character, each character with another character. So will there be a dot in this position? So matching character by character essentially means, again, in another sort of extension to that discussion, we discussed the concepts of window sizes and stringencies and so on, right? So here basically the window size will be 1 because we are matching each character by another character, and at every point where we find that there is a character match, we put dots. So basically, this A versus A junction point, we will put a dot, and then again we will just continue at every junction point in this grid. Whenever we are finding match, we will put a dot, and then at the end of this process, basically what we try to do is that we try to find diagonal runs of these dots, and these diagonal runs essentially will tell us which portion of sequence 1 will be aligning with which portion of sequence 2. So for example, if we compare this particular region, so here we see on the right hand side sequence we see ACG, and on the upper sequence we also see ACG. So this diagonal run is basically saying us that this ACG will be matching with this ACG. So we are finding an alignment there, right? Now that is again with window size 1 and stringency 1. Just to discuss, one way you can sort of manipulate how the overall view of the matrix will be in this particular process is to try to identify or manipulate the window sizes and stringencies of your records in a way that would sort of make the matrix slightly different. So what is the purpose of using, for example, a sliding window technique here? So let's say we have window size of 3 and stringency of 2. What that means is that we find 3 character or we match 3 character from sequence 1 with 3 character of sequence 2. And then if we find that exactly 2 characters are matching between these 3, so you do not need to have a exact match for all of, for all 3 of the elements in this window. But if only 2 elements are matching, at least 2 elements are matching, then you can put a dot there. For example, here ACG versus ACA, we see that the first 2 elements of both of these windows are matching. So that means that with a stringency of 2, we can put a dot where the junction point between these two window is, right? And again, we will slide these windows across by 1 element and essentially try to identify all of the 3 versus 3 window match with a stringency of 2, right? So after we, let's say, complete the process, wherever we find the matches, the entire dot plot will look slightly different here, right, than what we had if we used window size 1 and stringency of 1. So does anybody can anybody remind me what was the purpose of, let's say, increasing the window size or using the sliding window technique at all? So what purpose did that serve? You can write in the chat, or you can unmute also. So basically, you can find out the answer by looking at this plot versus this plot. We will see a lot less number of dots when we used or increased the window size, right? And what that basically means is that, let's say, in an actual alignment procedure where you have thousands of characters for sequence 1 and then thousands of characters for sequence 2. So if you match exactly every character with another character, so 1 character versus 1 character match, in that case, the entire matrix will be covered with dots, and essentially that would also mean that there is a lot more noise in this whole dot plot. So to reduce the noise and to find out the exact runs of diagonals, because the diagonal runs might not be visible clearly if there is a lot of noise basically in the dot plot. So to reduce that noise, we can use or we can manipulate the window size, and then at the same time we can use different stringency values to sort of identify which values for those will give us a better visualization essentially. So here was some examples. For example, so this was again a dot matrix for the amino acid sequence, and then in 3.4 we have dot matrix for DNA sequences. And again, we discussed also that if we have, let's say, DNA sequence versus DNA sequence dot plot, if we construct that, so there will be a more number of chances that the noise will be higher because only there are 4 nucleotides. So in 4 versus 4, the exact match will be easier than let's say if we were comparing with amino acid versus amino acid when there are 20 amino acids on each side, where that the probability of that amino acid versus amino acid match will be lower, right? So that was kind of quick recap of what happened. And in this particular dot plot, we are seeing that by manipulating the window size and the stringency values, we can go from something like something that looks like this to something that looks like this, which is a lot more interpretable. And what we can find in this particular stretch is that, okay, first of all, this is a diagonal run that is stretching right from the very beginning to the very end. So what does that indicate? If we have a diagonal run, which is the main diagonal of this matrix, by the way. So if that is sort of, from the very beginning to the very end, it is running. So what does that indicate? What does that say about this dot plot? Any idea? So at each dot that this diagonal, the main diagonal constitutes of, each dot will mention that there is a match on both of the sides of the sequences, right? So sequence 1 versus sequence 2, there is a character match. And since this is a continuous diagonal, that means that every element of sequence 1 is matching with corresponding element of sequence 2. So basically that means that this is the dot plot constructed using one nucleotide or one protein sequence, let's say, against the same sequence. On both axes, you have the same sequence. So that is why there is a diagonal that is running basically through the main diagonal of the matrix, right? And alongside that, we are also finding out that there are these sort of small stretches of diagonals that we can observe, right? Below this main diagonal and above this main diagonal also, which are basically mirrored on both sides, because this is the same sequence versus same sequence match dot plot. So what does these sort of diagonal stretches, a lot of them, right? We are seeing here at this very at this particular part, the one that I'm highlighting with the cursor. So what does this sort of pattern indicate? Anybody? Yeah, I see on the chat repeats. Exactly. So those are indicative of repeats. So when you construct dot plot for the same sequences on both axes, you can find repeats or repeating subsequences that are present within the nucleotide or protein sequence, right? So that is sort of another approach or another usefulness of constructing the dot plot here. So again, this was, for example, constructed with human LDL receptor against itself, and here they manipulated the window size. So here it is written in plot view window size of 3 and stringency of 7. So that sort of gave a clear visualization. Again, another example of why window size and stringency manipulation might be useful. Okay. Okay, fine. So another sort of stretch of concepts that Professor Brumia, for example, went over in earlier lectures also was categorization of some sort of functional proteins, right? For example, enzymes. So we also saw some of the similar types of questions which might come in your assignment also. So I have put some of those questions here as well. So let us start the session for the first question for today. So enzyme number 3.2.1.2, what will that belong to? So these basically indicate some sort of pattern, right? So this enzyme number that is represented here, what does that belong to? So you can write in the chat. Also, you can unmute. So I see some C answer in the chat. Yeah, so this is an example of a hydrolase. And what this enzyme commission number will tell is because basically the first element of this 4-element number, or the 4-element enzyme commission number, that will tell us about the class of the enzyme. So here, since the first one is 3, so 3 you can identify from this class number, that is of class hydrolase, right? So for example, here we have a very important reaction, ATP plus D-glucose to ADP plus glucose 6-phosphate, right? And here the enzyme that is involved, that is ATP glucose phosphotransferase. So the enzyme combination number will be 2.7.1.1, and the first number in this enzyme combination number is 2. So that refers to the class of the enzyme, which is transferase. Then the second number will represent the subclass, which in this case is phosphotransferase. Then the third number will be what type of phosphotransferase it is. So it is a phosphotransferase with a hydroxyl group acceptor. So that is classified as 1. So we'll put a 1 there. And for the fourth number, we have what acts as the phosphoryl group acceptor. In this particular case will be D-glucose. So that will be 1 again. So in this sort of way, you can find out the entire stretch of numbers for the enzyme function number. Right. So the overall classification here will belong to, it belongs to the classification of transferase class, particularly subclass phosphotransferase catalyzing the transport of phosphate groups to a hydroxyl group with D-glucose acting as the acceptor for the phosphoryl. Right. Another sort of concepts which might you see quite frequently in the assignments and exam questions is that there are certain inorganic ions which are required as cofactors for function of certain enzymes, right? So one of those important reactions, for example, is highlighted here, which is this one, right? CO2 plus H2O, and this particular enzyme is Ca, which is carbonic anhydrase, and then it catalyzes the reaction H plus HCO3 minus plus Cl, right? So this is the process where the CO2, the entire process is basically how tissue cells and alveoli are sort of exchanging gases throughout the circulatory system, right? So does anybody know what is the inorganic ion that is required for carbonic anhydrase? So again, you can use the chat or you can unmute also. So some these sort of like some of the most important enzymes that you see in biochemistry, like in biochemical reactions, you might want to remember their important cofactors if any are present, right? So this is one of those. So you can write in the chat and then we'll see. So the carbonic anhydrase requires the ion Zn2 plus, zinc 2 plus. Okay, and here are some of the important enzymes and their corresponding cofactors, ionic cofactors. So for example, Cu2 plus, copper 2 plus is required for cytochrome oxidase. Fe2 plus, Fe3 plus are required for cytochrome oxidase as well as catalase, peroxidase, etc. Mg2 plus required for hexokinase, pyruvate kinase, etc. Mn2 plus required for this arginase and so on. And then Zn2 plus also here we can see carbonic anhydrase is requiring Zn2 plus. Right. And again, some other information is also given in this table. For example, some coenzymes that serve as transient carriers. So you might want to look into it also if you are interested. Right. Okay. So those were some of the concepts from week 2, also like from earlier classes as well. Now, in continuation of the dot plots, so what is the necessity? We also went over this a little earlier also. So what is the necessity for, let's say, introducing this match, mismatch, and gaps codes? Why not just use a dot plot throughout, right? For any alignment requirement, why not just use a dot plot always? why do we need this sort of matches, mismatches? Why do we need to find out gaps, whether there are any, and assign scores to those? So it's because dot plot only gives like a visualization, right? But it's a kind of a crude visualization. However, to find out the exact alignments, we need a sort of rigid set of scores that will particularly be able to quantify whether one alignment is better than another or not. So again, in this exercise, what we have is that we have a particular set of matches, mismatches, and gaps. So we have 3 matches, 2 mismatches, and 1 gaps. And what we have been given are match scores, mismatch scores, and gap scores. So notice that we do not characterize the gaps in this question. So just a small numeric question this is. So what will be the total, what will be the overall alignment score for this set of matches, mismatches, and gaps? So I see 2, 7 answers in the chat, 3, I guess. So the others, you can also try to find the answer and write in the chat. So here, basically what you have to do is that you have to just numerically calculate how many matches are there, then how many mismatches are there, and then how many gaps are there. And you just add the total numbers to get the net alignment score, which in this case will be 7. So that was a simple one. Now, so the application of these sort of scoring systems, which we'll look in a bit with several alignment algorithms. But first, let us cover some of the other concepts, for example, PAM matrices and Blossom matrices and so on. So does anybody remember what do PAM matrices stand for? So what's the full form of PAM and what does it mean? So I see in the chat point accepted mutation. Correct. So PAM essentially stands for point accepted mutation. And this matrix was originally constructed by Margaret Dayhoff. And what Margaret Dayhoff and her colleagues, they basically did is that they compiled 71 groups of closely related protein sequences. And then they constructed the matrices. Now, the choice of closely homologous protein sequences is very important. And they set one criteria, which is that they used sequences with greater than or equal to 85% of similarity. And they sort of assumed some of the important assumptions that we'll see in some slides, I guess. So first of all, one of them is that the expected mutations in the closely related sequences will not change the overall function of the protein, first of all. And what we see is that, as per the characterization, is that the amino acid mutations that are observed in nature, those are called, or those are considered accepted by natural selection, right? And the derivation of PAM matrix is basically, firstly, the sequences are clustered using phylogenetic reconstruction. Then the mutation frequencies equivalent to a single PAM unit are constructed. And that will give the PAM1 substitution table. So one PAM unit, so can anybody remind me what does one PAM unit mean? So one PAM unit essentially indicate that if we are comparing, if we are, let's say, using 100 amino acid long sequence, then there will be 1% change in the amino acid positions or elements of the sequence. So that means that out of 100, one amino acid will be mutating. So that sort of way, right? In the same way, if we compare, let's say, or if we use the term PAM 250, so that will mean that of a 100 amino acid length, 100 amino acid long sequence, we'll see 250 mutations. So that means on average, one amino acid will be mutating 2.5 times. But that's only an average. So it does not mean that every single amino acid will mutate 2.5 times. So what it means is that some amino acid, for example, will be mutating at a higher rate. Some amino acids will not mutate at all. But the overall value that we will get or we expect to get will be 2.5 mutations per amino acid in case of PAM250. So that's sort of it. Now the derivation procedure is basically that the PAM score for particular residue pairs will be calculated first of all using relative mutability, which is the number of mutational changes from a common ancestor for a particular amino acid residue divided by the total number of such residues occurring in an alignment. So you have to consider or you have to calculate the relative mutability and you have to normalize the expected residue substitution frequencies, and then you will transform that into a log value. Basically here it is written that log base of 10. So that is why it is also called a log odds matrix. Okay. So how these log odds scores are calculated is basically the value is rounded to the nearest whole number and then the substitution matrix in the matrix it is put. And what this essentially reflects is that the likelihood of particular amino acid substitutions, right? So you can imagine that in the PAM matrix you will see. So let's say on the upper side you have the 20 amino acid sequences, and on the left hand side you have 20 amino acid sequences. So you'll see some values corresponding to each junction points, right? So what does a positive or negative value indicate in that sense, or in this particular case? If in a PAM substitution matrix at a junction point of two amino acids we see a positive score versus, let's say, a negative score, what does those indicate? Does the positive score indicate that it is more probable that this mutation will occur, or that it is unlikely by random chance? So we look at this concept as well. But first, let's say I ask what is the size of the PAM matrix we just discussed. So for the point accepted mutation for amino acid sequences that are put on the upper hand of the matrix and on the left side of the matrix, so what will be the size of this total matrix? So the size will be 20 by 20, right? Yeah, as I see in the chat. And interpretation of matrix score, what I just asked, is that the positive scores will indicate that the substitutions are occurring more frequently than anticipated among evolutionary conserved replacements, and the negative score will be kind of the opposite. So they correspond to substitutions that occur less frequently than expected, right? So we'll see an example in the next slide. And what the PAM1 matrix gives is that basically this is used for the closely related sequences, right? So homologous sequences, for example, what Margaret Dehoff considered was greater than equal to 85% of similarity they need to have, right? So for what? So what should we do for, let's say, more evolutionarily diverged set of sequences, which are sort of at a higher evolutionary distance than one another? So in that case, what it does is that it uses extrapolation. So from matrix multiplication, the other, on a higher evolutionary scale, those matrices, for example, if we consider PAM40, PAM80, PAM250, so those are considered constructed from matrix multiplication by multiplying PAM1 matrix with another. So that will give, let's say, PAM2, and then again 3 times, that will give PAM3, and so on. So this way, sort of, for example, PAM80 is generated by PAM1 matrix times PAM1 matrix. We have to do that 80 times, right? So PAM1 into PAM1, you have to do that 80 times. So that will give PAM80. And that way, you can basically extrapolate what will be the scores for the PAM matrix for a highly diverged set of sequences, which are evolutionary at a higher distance than one another, right? So, for example, these are some of the scored values here. So, PAM1, where we have the corresponding observed mutation rate, that is 1%. Sequence identity is, let's say, 99%, right? PAM30, you have observed mutation rate 25%, sequence identity is 75%, and so on. Okay. So, if I ask, okay, so we just discussed it, maybe I'll ask later. So, what is the difference between PAM matrix and Blossom matrix and their scoring? So we'll see that after we discuss the Blossom, I guess. So here, for example, this is an example of the PAM matrix. This is a PAM 250 scoring matrix. And on the upper hand side and on the left hand side, we are C, or on the lower hand on all of the sides. So the amino acids are mentioned basically. Basically on this axis, for example, you have all of the amino acids mentioned, and on the left hand side axis, you have all of the amino acids mentioned. In this particular example, they have grouped also the amino acids in several, I guess, smaller subgroups essentially. So, for example, based on their side chain chemistry, so you have some sulfhydryl containing groups, for example, STP, AG, cysteine, that is sulfhydryl, then small hydrophilic group, then acids, acid amides, hydrophilic basics, the small hydrophobics and aromatics, and so on. Now, if we take a quick look at this matrix, you will see that at every junction point, as I mentioned, there are certain scores written there. Okay. So, for example, if I ask from this particular PAM250 matrix, is it likely or not likely that, let's say, a glutamic acid, okay, so is it likely or not likely that a glutamic acid will be mutated to a lysine? So from this matrix representation, is it likely or not likely that we'll expect a glutamic acid to be mutated into a lysine? So you can answer in the chat. Yeah, as I see in the chat, it is not likely, right? So from common sense also, that kind of that the answer we can get, because glutamic acid and lysine has opposite charges, so they will sort of destabilize the structure if one negative charged amino acid was mutated to another positive charged amino acid. By referring to this matrix, we can also see, for example, this E versus K. Okay, so for example, here in this case, we have written 0. So for PAM 250 matrix, it says that it is neutral, right? I was actually considering in this example leucine. I kind of mistook the matrix notations from there. But anyway, so if we see, for example, E versus L, so glutamic acid to leucine, so we have a -3 score. So that means that kind of mutation will be not likely. Conversely, if we see some positive scores, so for example, here we have this 2 score, right? So this is aspartate to glutamine. So that gives a positive score. So that means we can expect that mutation sort of, right? So 0 scores will essentially indicate those are neutral. So it sort of balances out. For example, here we have a lysine to a glycine. So that substitution again has a negative score, so that is less likely. So if I ask from this matrix, name me two amino acids which resist mutation the most in this table. So in this table, which two amino acid you see that resist mutation through an evolutionary process? These two amino acids are very less likely to be mutated to some other amino acids. I see one answer, F to D. So if you take for example, where is phenylalanine, so that is phenylalanine, right? And then if we consider so okay, here we can see, so F to D, so the score in this case will be -6, right? So here on this side we have F, here on this side we have D, so the score will be -6. That's correct. That it is less likely. But what I was kind of asking is that which, let's say, two amino acids uh, that do not change into any other amino acid or that resist mutating to any other amino acids, or sort of like most of the amino acids, you can say, from this table. So they resist mutation just in general through an evolutionary process. So that's kind of what I was getting at. Yeah, so one answer I see, W, right? Tryptophan. So if you consider, for example, tryptophan with all of the elements, of course, here phenylalanine has 0, so neutral, Y has 0, and tryptophan with tryptophan itself is 17. But for all of the other cases that we will see, it has sort of high negative values, right? So tryptophan in this case will be resisting the mutation a lot, right? I see another one that is Y, right? So tyrosine in this case, you will see that of course it is resisting mutation a lot as well. So we are seeing -4, -2, -5, and so on. Some are neutral, for example, histidine in this case. Another one you can see, for example, cysteine, the first amino acid in this table. So if you compare, if you look at this column, you will see that cysteine also has a very high negative values against the other ones, except for tyrosine, right? So cysteine is another one that will change or resist changing to another amino acid throughout an evolutionary process. So you can sort of infer those sort of substitution rates and so on from this table. So that's the PAM matrix, right? So what are some of the assumptions that we talked about in PAM matrix? So this is another question that we just talked about. So the dimensions of a PAM matrix is 20 by 20 because on each hand side we have 20 versus 20 amino acids, right? So what are the assumptions? Some of the assumptions that are applied while constructing PAM matrices. Okay, we'll talk about that first. So let us solve this question first. Okay. So for evolutionary divergent sequences, which PAM matrix is commonly used? Try to answer in the chat and then we'll look at it. So I see one answer, positive score means mutation is more likely. Yeah, that's correct. That's the characterization of the scores or interpretation of the scores in the PAM matrix. Okay, that's correct. So for evolutionary divergent sequences, which PAM matrix are used? So I see some answer C's, which is correct. For evolutionary divergent sequences, TAM 250 matrices are used, right? So coming to the assumption point, we see that, so this will also let us see about the matrix multiplication, right? So what it assumes is that the change of the amino acids that counts as a Markov process. So Markov process means that it depends only the identity of amino acid at that current state, not not on any past history of which amino acid there was at this place. So in that sense, let's say if we talk about, let's say just an example sequence. So if we have, let's say, A, L, S, T, some sort of sequence like that, right? Dot, 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 dot. So if this amino acid sequence was, let's say, mutating first to, this alanine was mutating to threonine, let's say, right? So threonine, L, S, T at this position. And then this threonine was again mutating to serine. So you have something like this. And then in the end, again, this serine is mutating to alanine, right? So throughout an evolutionary process, we are seeing that in this particular case, the alanine is being mutated 3 times, right? So first it is alanine to threonine, threonine to serine, and then serine to alanine again. And each of these states, or each of the step of the mutations, Markov process assumes that only the current state will be affecting the mutation to the next step. So it does not matter. So for example, here we have an alanine. So it does not matter that at this particular at this particular place, we originally had an alanine. It does not matter that there was this sort of element present at this particular position. Only serine to threonine alanine, serine to alanine mutation is considered what's important for this particular change to occur. And again, it also assumes that the mutations will accumulate gradually. So let's start this one. So mutations will assume gradually or accumulate. Sorry, mutations will accumulate gradually, and it can be reversible as well, as we saw that if at the initial position there was an alanine, so after multiple times of mutations, we can again expect to see alanine in that place, right? And the distant related proteins, so matrices for those can be extrapolated using matrices used that are from close that are created using closely related sequences. So for this case, we saw that PAM1 matrix, let's say. So PAM1 means that out of, if we have like a sequence length of 100, so for this 100 length amino acid or nucleotide, in the case of, like, if we sort of try to apply this idea to nucleotide as well. So in this case, amino acid. So the amino acid for every 100, we will see one amino acid changing. In case of PAM250, that will be for every 100 amino acids, we will see 250 amino acid changes. So that means each amino acid will be mutating to 2.5 times, right? And that is sort of what it means, because you might get confused sometimes that how can each 100 amino acid mutate to 250 different times, or let's say 100 amino acids can, how can they mutate to 250 different states, right? Or different element mutations. But that is because there are a lot of intermediate mutations here as well. So as we saw, one particular point or one particular position can mutate to another, let's say, amino acid multiple times. So that is all averaged out and giving this code at the end, right? And here also something else is assumed, is that for each evolutionary interval, the frequency of change of amino acids, they are the same for short-term calculations. Okay. So again, for the distantly related sequences, we have to use matrix multiplication to sort of extrapolate what will be the scores for those sequences, right? Now, for this particular lack of statistics for divergent sequences, the Blossom matrix was developed, which stands for the blocks amino acid substitution matrix, and they are essentially created from direct observations. Okay, so direct observations on blocks of subsequences from, let's say, set of sequences of every possible amino acid substitutions in multiple sequence alignments. So from that, the Blossom matrix values are calculated. So in this one, for example, more than 2000 conserved amino acid patterns, those are represented by blocks of less than 60 amino acid residues, which were used to calculate the Blossom matrix. So less than 60 residues blocks, and in this particular one, the extrapolation is not done, but actual percentage identity values for sequences in this construction are used. So for example, in Blossom 62 matrix, we will see that indicates an average sequence identity of 62% or less, you can say, right, for Blossom 62. And so as you might assume from that is that the order of the number for the PAM matrices, those are opposite to the order in the Blossom matrix. So in reverse order. So in the case of the PAM matrix, we saw that the lower the PAM number is, so we can use that for closely related sequences. The higher the PAM number is, we use for more divergent sequences. In this Blossom matrix, the higher the number is, we can use that for closely related sequences. The lower the number is, that will be for more divergent sequences, because you can sort of get from this idea that if we, let's say, use a lower Blossom value, so the level of sequence identity will be that value or less, right? So if you, for example, use a 20% sequence identity or less, so you can use that to construct Blossom 20, and that will give, obviously, because the sequence identity is lower, so that will mean it is more evolutionarily divergent. It is good to use that for more evolutionarily divergent sequences. And again, as in the case of PAM matrix, so here also a base 2 logarithm is used. So in PAM, we saw that base 10 logarithmic value was used. Here, the base 2 are used, right? And for the values or the scores in the matrix, so positive and negative values again indicate that substitutions are occurring more frequently or less frequently, respectively. So those are the concept for Blossom matrices. And just to give a quick example, again, in this case, we see the same 20 by 20 matrix. And for each of these junction points, we see some sort of scoring, right? So again, you can use that to infer some data about, infer some scoring points that can be used in alignment procedures, right? Okay. So
excuse me, I have a doubt. Can you go back to the previous slide?
Sure.
So here, for example, if you talk about the top left corner, C versus C mutation is 9. So does that mean C is coded by multiple codons? So the change in codon, like ACGT होता है, तो जो 3 कोडॉन है उसमें से चेंज हो करके, बट इट इज कोडिंग अगेन फॉर सिस्टीन ओनली, मतलब दैट 3 न्यूक्लियोटाइड कोडॉन्स, एंड डज इट मीन दैट, और डिड आई इंटरप्रेट रॉन्ग?
So in this particular case, even without thinking about the concept of codons, it's basically that C versus C, because it is the same thing, right? So that is why there is a positive score. So,
but if it is C versus C, so it will not be mutation, no? It will be the same one.
Yeah, it is not the mutation, but because here we are representing a matrix, right? So for the construction of the matrix, every element is used here. That's sort of the concept. And if you talk about, for example, then why, let's say C versus C has a score of 9, but then T versus T has a score of 5, and then A versus A have scores of 4. Because all of those should indicate that the same amino acid is remaining same, right? Now here the concept lies is that there are different intermediate mutations that are occurring. Now what you said, it could be very true. Possibly the reason is that, let's say, one amino acid to another amino acid, this sort of score we are getting 4. So that means that some change in mutation or intermediate mutations are occurring for alanine more, right? So for example, in cysteine, it is occurring less. some sort of intermediate mutations. So that is why it is having a high positive value. And for alanine, the frequency of these intermediate mutations or the intermediate steps, for example, those are very less, right? So it could be very much possible that due to, let's say, the presence of multiple codons or something, that is sort of preventing this change in amino acids from one to another, you can say, or sort of preventing the rate of intermediate mutations in between. You can think of it like that, maybe. It's possible.
Okay, thanks.
Yeah. So in this question, let us try to understand something that we also discussed earlier. So which of these would be more suitable to use for closely related sequences, which are evolutionarily very closely related? We have been given numbers for the PAM value as well as the number for the blossom value, right? So try to answer in the chat, and then we'll look at it. So you see some answer A in the chat. And yeah, so the lower number for the PAM matrix, that will indicate, let's say, more suitable for closely related sequences, and the higher number of blossom matrix, that will again indicate uh, more suitability for closely related sequences as well, right? Now in sequence alignment, so we are often not only given the alignment scores and so on, but we have some associated statistical values, right? So values that will let us judge some sort of, or infer some sort of statistical parameters or understanding of the sequence alignment. So one of those are E values. Now, which of the following E values will be the most significant for an alignment? Which of these E values will be the most significant for an alignment? So again, try to answer in the chat, and then we'll look at it. So here in this particular case, the lower the E value, that will be the most significant for an alignment. Now, what this E value is basically it is the probability that the resulting alignment from a database search, they are caused by random chance, right? Another way you can put it is that it's the expected number of hits that are as good or better than the observed alignment if the query and the database are random with respect to one another. Okay. Now, typically we consider a threshold value for E. If it is less than the particular threshold, then we will say it is statistically significant. For example, the most commonly used is less than 0.005. Okay. And this is again equivalent to selecting alignments with scores above a certain threshold. But it's sort of states about the statistical significance of it. So what it basically is, if I can give like a small visual demonstration. So let's say we have some sort of alignment score on this axis, right? So let me just write it down carefully. So let's say on the x-axis, we have some local alignment score, and let's say on the y-axis, we have number of sequences, right? Now, in an ideal sort of situation, what you should get is that, so if you have, let's say, this set as a threshold for a local alignment score, so for unrelated sequences, which there will be a lot of, so for an ideal situation, you should see something like this, okay? And then let's say, for example, small hump here. So this is, essentially indicating that the alignment for the related sequences. So these, let's say, we are showing related sequences, and these, let's say, we are saying unrelated sequences, right? So ideally, we'll see that only above the threshold local alignment score, we will get the related sequences, and then below the threshold of the local alignment score, we will get only unrelated sequences. So that is sort of an ideal case. However, in actual alignment procedures, this does not happen. Now, what ends up happening is that, let's say again, we consider another graph. So if it is local alignment score, and if this is the number of sequences, and let's say this is the threshold value, what we will see is that if we construct the plot, let's say it has let's say it has a distribution like this, right? So let's say these are related, and then this is unrelated sequence that we are getting local alignment scores for. So in actual cases, we will see that the threshold value do not, clearly distinguish between unrelated sequences and related sequences that we'll get for the as the hits of the alignments, right? So there is some sort of overlap between the unrelated sort of distribution, sequences distribution, and the related sequences distribution. Now, what e-value particularly describes is that the expected number of hits with a score above the threshold if the query and the database are unrelated. So what that essentially means is that it provides an estimate for this particular region. So for the number of false positive hits. Okay, so this will be the number of, let's say, false positive hits, and then the e-value will give an estimate of that. And the lower the e-value, so that will basically mean that the related sequences and the unrelated sequences are clearly distinguished, right? So that will give a better statistical significance essentially. And in practice, for example, so let's say for three different alignments, so we have let's say one have the value of 0, another has 0, another has 0, and then another has 0.3. Okay. So for this particular one, let's say 0.3, and let's say it had, for example, corresponding scores. So let's say the alignment scores was like 677. Let's say it was 676. Let's say it was 590, and let's say it was 42.7 or something. So here, what the 0.3 value will indicate is that a score of 42.7 or better can be expected to occur 3 times in 100, 3 out of 100 times by random chance. So a score of 42.7 or better is expected to occur basically out of 100 times, 3 times out of just random chance. So you can just think of it like that: if out of 100 times, 3 times I am getting this particular score, so it might not be the most reliable one, right? Uh, so again, that is sort of so okay. So that was 0.03. So that will give 3 times. 0.3 will be a lot higher, right? 30%. So if it had like 0.03, so we can say that out of 100, 3 will be the occurrence in random chance, right? Occurrence by random chance. So that would not be the most statistical significant thing. So that is why kind of less than 0.005 of E value are considered in alignment procedures. So E less than 0.005, those are considered significant. Okay. So that was something about some statistics about alignments, right? Now another concept that we saw in the week's lectures were calculation of conservation scores. So they basically indicate that for a multiple sequence alignment, let's say at a particular position, what position 4, 5, 6, something like that. What is the conservation score for that particular element position or particular slot, you can say, right? So several methods were introduced. One of them, Professor Tomiha mentioned, are unweighted entropy-based measures, right? So here in this question, we have, let's say in a particular multiple sequence alignment, one position is occupied with the same amino acid residue, right? So if there are 10 total sequences in this alignment, let's say we're talking about position 2. So position 2 only has one particular residue for all of these cases. Or, I mean, the same residue for all of these cases. So if there was alanine in position 2 in sequence 1, sequence 2 to 9 also has alanine in position 2, right? So in that case, what will be the conservation score from unweighted entropy based measures? So here is a handy formula if you want to calculate that. So it says uh, summation of Fa of I times ln Fa of I. Okay, so could you calculate the score and then give the answer? So here the calculation is kind of uh, let's say using a formula. So Fa of I times ln Fa of I. Okay. Now, if a particular position is occupied in the same by the same amino acid sequences, so what will this Fa of I value indicate? This Fa of I that we are again multiplying by the logarithm of that same one. So here the frequency for that particular amino acid or the frequency of occurrence at this particular position, since only one amino acid is present there for each sequence, so you only need to calculate for one particular amino acid. What will that be? So that will be 1, right? Because only one amino acid is present. So let's say there are like 1, 2, 3, 4, 5, 6 positions, and each of them had alanine at this particular position, right? So the conservation score will be calculated in a way where we have to identify the frequency of alanine at that position. Now here, since all of the sequences have alanine, so let's say for this particular position, the number of alanine divided by the total number of sequences, right? So that will be 7 by 7. 1, 2, 3, 4, 5, 6, 7. It was 7 sequences. That will be 7 by 7 equals to 1, for example, in this particular case. So this ln FAI, so that will essentially mean ln 1, right, which will be 0. So multiply that by again Fa of I 1, so it will give you a score of 0, right? So in this case, the net score will be 0 because we only need to calculate this for one particular amino acid, which is alanine. If there are, for example, let's say other amino acids in this case, so let's say if there was alanine, and then in one sequence alignment we see threonine present there, then another one we see serine there. So for that one we needed to calculate 3 times: one for alanine, one for threonine, one for serine. Let's say in another one if serine was present, right? And then ultimately we have to do summation of all of these calculations. But here we didn't need to do that because only alanine was mentioned here, right? Okay. Now, for the alignment processes, so there are 2 types of alignment that are covered, right? One is global alignment, another one is local alignment. Now, what does local alignment useful for? What is it useful for? So that question is asked here. For overall efficiency, number of gaps, number of mismatch, or something like similar subsequence. So what would the function or the purpose of local alignment be? So you can answer in the chat. So I see one answer D, I guess. Global alignment is basically aligning in a way that the entire sequence is considered, or the alignment for the entire sequence is considered, whereas local alignment is about particularly identifying smaller stretch of subsequences which could be similar for the sequences, right, that we are aligning here. So essentially for identifying similar subsequences in this case, which could be useful for let's say motif discovery, right? Or again, another tool that we have heard of, BLAST, for example, when you use to find similar sequences from a database, right? We give query from a smaller fragment of sequence. So in that case also, it does not make sense to try to do a global alignment, but in this case, local alignment algorithms are used, which we'll look at it in a bit. The algorithms related to global and local alignments and so on. So again, uh, so in global alignment, let's say, where we try to align the entirety of sequences, so the value of each element in the matrix is assigned using some sort of scoring scheme. Now, what does that scoring scheme involve? So this is particularly asking for global alignment, not for local alignment. Okay. So in this coding scheme, which among these are involved? Now, in this particular case, the answer is given as B, but I see some answer D in the chat, for example, using 0. Now, in a way, we'll see that when you start the very first calculation using the, let's say, top left corner, we put a 0 there. But overall, the importance of 0, that was not considered. So this is also one of the earlier iteration of the class. So in last years, last to last years, so where this sort of question was present. And then 0, we will see that it is mostly important in the case of local alignment. Okay. But for the main scoring scheme, even though we start from 0 for the top, bottom, left position in global alignment algorithms, we'll see that the score is designated using match or mismatch. So that's a substitution in the case of mismatch, as well as indels. So these are insertions or deletions. So we'll see the example of the scoring for global alignments just in a bit. Okay. Now we earlier calculated or we talked about e-values, right? So those are sort of the overlapping section between unrelated sequence and related sequences in a local alignment plot that we saw earlier, right? This code versus number plot. Now here we have given some parameters. So when aligning a query sequence which has 100 residues against a database of total of 10,012 residues, so for HSP, the p value is given 1 times 10 to the power minus 20. So what will be the e value? And okay, so could you try to calculate it first, maybe? Okay, so we'll just walk through this question. So in this particular case, the e values can be calculated using 3 different elements. So one is m, n, and p, as it is designated here. So m will be the total number of residues in the database, or the sum of the length of sequences in the database. Then n will be the number of residues in query, and then p will be that probability of the HSP alignment being due to the random chance. Okay. So in this case, m times n times p, that will give the e value. So it will be essentially 10 to the power 12 times 100 times 10 to the power minus 20. That will give you a value of 10 to the power minus 6. So this sort of numeric questions you will encounter as well. Now, so let us go forward with the algorithms for alignments, right? Now for global alignment, excuse me. So for global alignment, which common algorithm is used? Can anybody mention in the chat? Or you can unmute also. So for global alignment, we are talking about which algorithm is used. Yeah, I see in the chat it's Needleman-Wunsch, right? So that's the algorithm used for global alignment. And let's say we provide an example for the global alignment procedures. So on the left-hand side, we have sequence 1, which is indicated as I here. On the upper side, we have sequence 2, that is indicated by J here. And for this alignment, we will consider max score of 1, mismatch score of -1, and then gap score of -2. Okay. Now, the first row and column of all of these, when we initiate the process, so the first row and column, all of them will be indels or insertions and deletions or gaps. Conversely, you can say. Now, what we do is that starting from the top left one, the gap penalty will accumulate throughout the horizontal and vertical steps. Okay. Now, what that will mean is that, let's say we start with 0 at this particular position. So when you move to the right one step, so that will essentially indicate the vertical or horizontal jumps. That will indicate one indel. So for indel, we use the gap penalty or the gap score. So for example, in this one, we will score the grid with -2 because we have a gap score of -2. Now this gap score will accumulate over each jumps. So again, for the right hand side, we'll add another gap score and give it -4. So again, we'll give it -6, -8, and so on. For the column here, so for the after starting from 0 again, we will get -2 first, and then -4 second, and then -6, and then -8. So that is how the calculations are sort of initialized. Okay, in this one. Now, what we have to do in the case of global alignment is that, first of all, we have to fill up these blank spaces where we do not see, does it jump because it is a new gap? Yeah, it is a new gap. So remember in the concept of dot plots also, when we used dot plots for visualization. So what we had is that so if you consider something like this, so remember, so let's say this was sequence 1 and then this is sequence 2. So let's say if we had in the dot plot something like this. So we had a diagonal run, and then let's say another diagonal run here. Okay. So there was some gap present between these two stretches of diagonals. And in the dot plot, what it would mean is that, so right up till this point from sequence 1 and sequence 2, every element was aligning. However, in this particular position, so the elements of sequence 1 sort of moved a short stretch after, right? And after that again, the alignment started from this one right up till the very end, right? So if you look at it this way, now this sort of stretch of sequence will mean that this sort of gap present here would have meant for the dot plot is that sequence 1 has some sort of insertion for a, let's say, small stretch of sequence in the middle, right, where the element for sequence 1 moved a certain bit and the 2's remain constant in this place. Now again, the same sort of concept is here. So for each grid that will go, so if you consider, let's say, character by character in dot plot versus this one, so here the concept is basically is that instead of putting dots to characterize each matches or mismatches or gaps, we'll put, instead of dots, we'll put scores. So that will allow us to better quantify the result, right? So when we accumulate, let's say, when we go from -4 to -2 to -4, that will essentially indicate that each time we see a stretch, let's say like this, in the alignment. So when the alignment path sort of moves in a vertical or horizontal direction, so that will mean that there are gaps present in this case. And each time a new gap is there, since we have to give the gap penalty for each of these gaps, we will just accumulate this scores, right? Now the main concept where it is, where it is applied in the calculation. So we can initialize the topmost and the leftmost, columns and rows and columns here, but then we need to fill the entire of the rest of the grids that are present, right? Now for this calculation, we have to sort of trace some paths. Now For the vertical and horizontal lines, we only had to calculate, let's say, from this to this one and then this to that one, right? So by accumulating gaps from a predecessor, you can say in a way. For this cell, if we want to calculate, so you have to consider three different paths where the score can be taken from. One would be this diagonal path, another would be right from above, another would be right from left, right from the left on left element here. And what we have to do is that we have to calculate for each of these paths, and then the best score that we will get, we will consider that path for the path tracing, which we'll see later. So if for this small grid, if we let's say identify the top left one by s of I minus 1 j minus 1, and let's say this one is s of I minus 1 j, this one is s of I minus 1, sorry, I j minus 1. So from all of these points, we have to sort of get to this Sij point for the current grid for which we will calculate the value of. Now it can be calculated from three different paths. So this diagonal path will indicate that there will be a match or a mismatch. And as we discussed earlier, the horizontal or the vertical paths, those will indicate gaps, right? So the value for the current grid that we will calculate, or the Sij, it will be the maximum score among the three possibilities. What are the three possibilities? Firstly, one possibility is that we will trace this path, which is S of I minus 1 j minus 1. So this will consider a match or a mismatch. So a match or a mismatch will be considered by this. And then the second one will be, let's say, S of I minus 1 2j. So this will be this path. So that will again will be considered as a gap penalty. And then the another one is this the horizontal one. So S of I and j minus 1. That will also be considered as gap penalty. Now we have to calculate that and then identify what is the maximum value among these three. So if I ask, let's say I remove this. Okay. So if I ask, if we let's say take this one. So for the S I minus 1 j minus 1, if we count or trace the path for this particular cell, so what will be the score uh, for this grid? So if we trace the diagonal path, what will be the score for this grid? We have been given max score of 1, mismatch score of -1, and gap of -2. So since this is a match, as I see in the chat, so since this is a match, because it is a aspartate versus aspartate, right? So it is D versus D on both hands. So we'll have to give 0 plus 1 equals to 1, right? Yeah, so that was 0. So let me just remove these ones. So the top and the bottoms, top row and the left-hand side columns, those will be indicative of accumulating gap penalties. And in this particular case, so if we go from the diagonal, if we trace the diagonal path, we'll see that it will be 0 plus 1 because we have a match equals to 1. If we jump from the I minus 1 j position, so this vertical arrow, if we trace, what we will see is that since this is considered a gap penalty, so we will see the starting value, which is -2, accumulating with another gap penalty will be -4 total. And for the left hand side to right hand side, we will also see the same thing because we are starting with a -2, and then we are accumulating another gap penalty. This will be equal to -4, and of course the maximum of these three will be 1, right? So we'll put the value 1 there. Okay, for this one. Then what will be the calculated value for this grid, the one that is just right hand side of the one valued grid? So what will be the calculated value for this grid? So again, you have to consider three paths, right? So one is diagonal, which is match or mismatch, and then the vertical ones and the horizontal ones are gaps. So let's try to find out. I see one answer, one in the chat. So for the diagonal path, since this is a mismatch, right? So this is P versus D here. Since this is a mismatch, so we have to add a score of -1, right? So this will be -2, -1, -2 because we are starting from this particular position in this case. So -2, -1 will be -3. Okay. And then for the horizontal calculation, we'll see -4 -2 equals -6. And then for the, sorry, that is the vertical calculation. And then the horizontal left to right tracing, we'll see 1 -2 equals -1, because again, it is a gap. So the maximum of this is of course -1. So we'll put a value of -1 there, right? Now using this process, we have to fill out the entire matrix basically. Okay. Now upon filling the entire matrix, then you have to identify, okay, what will be the alignment. Another important concept, or important process that I did not kind of mention, is that whenever you are reaching the maximum or reaching the proper value for a particular grid, you have to kind of put an arrow from which direction this value is obtained from. So if it's obtained from the upper hand side, you put an arrow in this direction, which will essentially which will be able to allow us to trace the path to get the alignment, as we'll see later. So for example, in this particular case, after filling the entire matrix or entire grid, so what will be the net alignment score for this DPLE versus DPME, in a Needleman-Wunsch algorithm? So what will be the optimal alignment score or the global alignment score? So for this global alignment grids, the value that we see at the bottom right, so in this case that is 2, so that is the net score or the net alignment score. Okay. So that was the Needleman-Wunsch algorithm. And what I just talked about was that you have to sort of put, when you obtain every new value, you have to sort of put an arrow which will let us know from which direction this value is obtained from. So which calculation basically gave us, or which path gave us the max value for that grid, right? So in that way, what you can do ultimately is that you can look at the net alignment score, and then you can trace it back to the original position. Now what that will give us is that will give us the best alignment, right? So in this one, if we start to trace back, so for example, let's say we go 2 to 1 in this way, and then in this way, then in this way, then in this way, here, here, and then here. Let's say that is the traced path here. Okay. Now what we have to do is basically, so the first two, let's say, sequences. So if you consider this one, so in earlier case it was considered 2 and 1. So if we just consider this as 1, if we consider this as sequence 2. So of course what is happening here is that the first two, let's say like this. So the first two elements are matching. And then right after that, we see the presence of a gap. Okay. And this presence of a gap, what it is doing is that for the number 2 sequence, it is moving the element one place forward. Okay. So here we'll just add that element here, C, and then in this particular position for sequence 1, we'll put a gap here. And then again, we see for T and G, so for TAG, again, it is a match in this case. So TG is matching here also. And then right after that, we are seeing another gap here, right? So in this one, what is happening is that sequence 1 is moving again 1 place forward. So we'll just add a T here and another gap here. And then again, in the final case, we see a TA here, right? So TG and then TA here, TA here. So that is sort of the alignment that we are getting from this traceback. Okay. So this is saying that the sequence 1 had gap at this position, sequence 2 had gap in this position, basically, right? So you can find the alignment this way. Now, does anybody see some sort of another inference that we can get from this alignment? So I have identified the traceback, and that traceback gives us particular alignment, right? For alignment score of 2. Now, does anybody see whether we can get any other inference from this scoring and the traceback here, or just from this entire matrix? Anyone in the chat? Yeah, I see one answer in the chat. It is 2, 2, 1, 2, 3. Yeah, exactly. So remember that we did the traceback from here to here, right? But another way we are seeing that we are, arriving from this grid to this grid is that by tracing this path. So the 2 to 1 to 3, this traceback is also possible for this alignment, right? And what that would look like, basically something like this. So if we say we are numbering the upper one and then the left hand side one 2 as well, but in general the left hand side is numbered 1 and the upper one is number 2, but anyway it's kind of just the matter of numbering. So if we do it this way, so what will happen? So the firstly we had CA again match, CA again match, and then at this position we see a gap here, we see the element C here, and then right after that TG and TG. So up till this point, it is similar, right, to what we obtained earlier. However, now we have a new path to follow, sort of, right? So in this way, the path is following. And again, for this one, we are seeing that TG and the next element, so T, those are following a diagonal line. So that means that we have to sort of put whatever element is there at the particular position, at the particular place, right? But for immediately after that, we are seeing again the presence of another gap. And in this gap, we see sequence 1 is moving forward sort of one time. However, sequence 2 is staying at the same place. So here we can see, we can consider this T being present here. That was sequence 1. So just erase it. Yeah. So for sequence 1, we see another T is present. However, in this particular place, sequence 2 remained, the element remained at the same place, right? So T versus a gap we can see here, and then the last A will be matching. Okay. So both of these two alignments are possible in this case, and both of them give the optimum score of, or the net score of 2. So that can also happen where basically you get the same score for multiple alignments in two sequences, right? Okay. So let's just erase it. Yeah. So these two alignments you can get from this set of two sequences, right? Now, of course, in practice, actually, this algorithm will be more complicated, where the scorings will be, again decided by, for example, the sort of substitution matrices that we looked at earlier. So from them, and then the gap, the consideration of gaps might be different. What will be the gap score, gap penalty score, and so on. So those will influence the total overall score, and consequently, those will also influence the alignment that you are getting. For example, if you change, let's say, the gap penalty points from -2 to -3, you might see some other optimum alignment that is occurring between these two sequences. So the change of or the inclusion of scoring for matches, mismatches, gaps, and so on, or any particular set of mutations. So those are also important to the overall alignment and the alignment scores. Okay, so let us try to calculate this one. So here we have I sequence 1 on the left hand side, and then J sequence 2 for the upper side, right? So we have been given ATTGC versus AGTTC. So for this one, again, we are considering match of +2, mismatch of -1, and then gap of -2. So we are putting a bit of a more importance in the match here. Earlier we saw using match score of +1. Here we have match score of +2, and the mismatch is same, -1. So the gaps are also same, -2, right? So could you calculate the value for X in this alignment matrix? So the procedure is the same. You just have to calculate the value. So if I may initialize the upper row and the left hand side column, so it should look something like this, right? 0, then we are accumulating gaps over each step. So -2, -4, -6, -8, -10, and in this case the same, 0, -2, -4, and so on. For each grid, again, we have to calculate 3 different paths. So one will be like this, another will be like this, another will be like this. Okay. So if we start the calculation, for example, for this grid, what will this be? So for the diagonal value, we will get, since this is Aladdin versus Aladdin, so we'll get 0 plus 2 equals to 2. For the vertical one, we'll get -4. For the horizontal one, we'll get -4. So that should be 2. Okay. Then you have to calculate this grid. So again, for the diagonal, what we will get? We will get -2 plus 2 equals 0. From the vertical path, we will get -6. From the horizontal path, we will get 2 -2, right? I think, yeah. So in the same way, you have to calculate sort of. Yeah, I think I made the mistake here because no, it should be mismatch, so it should be -3. So could you calculate it out? So if you follow the procedure like this, just by filling the gap in this particular case. So in the next one, we have to calculate this one. Then when you have these three, these three values at the proper places, we can calculate this one, and then this one, and then finally we can calculate the value of x. So if you go through the procedure, you should see a value of 2, as I see in the chat also. So the same procedure, right? Now, we discussed about global alignment. However, there is another, a sort of alignment that we can do, which is called local alignment, which earlier also we saw in questions, right? So the local alignment is basically trying to identify smaller stretches of subsequences between two sequences, right? And the distinction between the global alignment and the local alignment is that, so that also relate to one of the earlier questions where we talked about which sort of scoring was considered. So for global, we saw that match score, mismatch score, or the indels or gaps are considered, right? For the local alignment, again, we are introducing the concept of zero. So this is sort of a modification of the Needleman-Wunsch algorithm, where the point is that, again, we will start tracing from the very upward position from the first node, or let's say. So we'll start with 0. But here the concept is that every time you see a negative score for each of any of these grids, so instead of putting the negative score, what we do is that you put a 0 here. So this basically allows any node to start at 0. Okay. So node calculations for a particular cell that cannot be negative. And then for that, again, you have to do a traceback, as we saw earlier. But that traceback is kind of also different in a way that you have to start from the optimal alignment value. But here it is not the bottom right cell. The optimal alignment value for the local alignment is not the bottom right cell. It is essentially whichever cell has the highest value in this grid, that will be the optimum alignment score, or the alignment score, net score basically. And you have to start the traceback from there. So that's the sort of fundamentals for the local alignment, right?
Excuse me, what decides the value of match, gap, or mismatch? Like in the three examples, you have given different values for match or gaps or match. So who decides, or what factors decides that?
So the factors, so these are basically very simple numeric problems. And the factors that are deciding is that throughout an evolutionary process, let's say, one amino acid staying the same will be preferable, right? So there will be the mutation rate will be lower. So in that sense, we are seeing that the match values or the match points are preferred. So that is why we give a positive score here. For the mismatches, again, substitutions or mutations can occur, which will be less favorable to the evolutionary process again. So those are slightly lesser favored. And earlier, we also saw that for the gaps, whenever there is a present of indels, insertions or deletions, so those are even less favorable throughout this process. So again, less favorability means that we have to give it a higher penalty. So that is why we introduce gap penalty. Now, the deciding factor among all of these does not come simply from the simple numeric examples. So, for example, every mismatch is also considered differently. So, what we saw in the case of constructing those matrices, right? Prime matrices, blossom matrices, and so on, where the matches will be scored in certain way, and the mismatches will also be scored in certain way based on the, let's say, observed probability or the relative mutability and the calculation of the log odds score from that. So that is the concept of M matrices. And then the blossom matrices was also constructed using the evolutionary, conserved regions, conserved blocks of sequences, right? So it's not the case that the match score will be +5, the match score will be +1, the match score will be +2. Those scores in practice are calculated using those particular matrices that we talked about earlier. And for the gap, again, what fits, what suits the best. For example, if we had an origination penalty, we'll give it again higher penalty because throughout evolution it is not favored at all, right? That there is an origin of a new gap and so on. So in this way, sort of the values are used, let's say, practically in actual alignments. Here, it is just a numeric problem using simple values for just conceptual purposes. That a match is favored, so we give it positive value. mismatch is unfavored, so sometimes we give it negative value. In this case, mismatch is considered 0. So that's just different sort of numeric scales that you can consider. But actually, those are counted or calculated from the substitution matrices in actual use.
Okay. Another question that I have is like in the previous example where
Yeah.
you have drawn a gap and in the previous one.
This one?
Yeah, this one only. So
Yeah.
amongst this, which two alignment is better, or I mean, if it comes in the question in the exam, so if I write either of the two will be okay, or
Yeah.
does this also have some preference? Ki this one is better, or
No, using this particular scoring scheme that was discussed here, so both of them are counted as equal using that scoring scheme. So it can happen also, and it also depends on the particular scoring that we consider. So the gap score of -2, for example, the match score of +1, mismatch of -1, and so on. So it also depends on the scoring. But whenever you are given a particular scoring, if you are seeing two alignments that result in the exact net score of the optimal alignment, then those two should be counted as equal. Okay.
so in exam we have to write both the sequences, or any one will go, and this is only for the exam level, but otherwise in research purposes or matlab, which one is considered better?
Now, usually for the research purposes, you will use more complicated scoring system. So the algorithm will sort of decide that which alignments are better. Okay, in a way, if you see the uh, sort of same scores, so those can be considered again equally. But that will have to be judged on case by case basis. For exam purposes, again, these should be counted as equal, because this in the simple numeric questions there is not any complex considerations of particular matches, particular mismatches, and so on. So whatever you are getting from the traceback from the optimal alignment, you have to count them as equal here. And for the research purposes, I guess you can say that it should be case by case basis. You have to identify. Usually, the software or the algorithms that you are using will judge those for you anyways. Okay.
Okay, thanks. And this can be used only for two sequences. For multiple sequence alignment it cannot be used, I guess.
For those again, you have to do it multiple times, I guess. So I see another one where we can access the recordings. So the recordings will be shared through the YouTube links. I think you have already received or you have a tab on the portal where the spreadsheet is shared, right? So the recording for the YouTube link should be shared there. Yeah. So for multiple sequence also, like in case of BLAST, what we see, so when you search a database using local, sort of using the local alignment calculations, so there also these sort of concepts apply. But in BLAST particularly, we see some sort of pre-screening is done based on a particular set of characters that are matching. Okay, so we kind of call it KMRs. So the number of K values that we will give, so it will construct that sort of subsequences from the entire sequence that you are giving, let's say as query, and then it will find particular sequences that are matching with this particular KMRs, and it will consider that as sort of initialization seed, and then proceed the alignment through that way. So it will count it will consider the alignment for all of these sequences, and then based on the best results, it will give, okay, this alignment is possible, or this is the hit that we are finding, that sort of thing. So the best alignment score
Okay.
hits will be reported in that sense, right? Because anyway, for each
Okay.
alignment, you have to use like sequence 1 versus sequence 2, and then if a third sequence comes into the picture that you want to align with the query, then you just have to do sequence 1 versus sequence 3 alignment and so on. Yeah. So the concept of the Smith-Waterman algorithm was that instead of let's say putting negative values, at the positions where the negative values are calculated. Instead, we put zeros there. So at each step of the calculation, we sort of put a value of 0, right? And in this case, if we try to calculate the value of x, so again from the diagonal section, we see a mismatch. So for the diagonal calculation, so the calculation process is same as the Needleman-Wunsch algorithm. So there is a mismatch. This is A versus T, right? So this score will be 0, right? And then from the vertical calculation, we see again we will have 0 minus 2, which is gap score, so that will be minus 2. From the horizontal path, we will see that will be 5 minus 2 equals 3. So in this particular case, the alignment score will be 3. Now let's say that we had an alignment score, if we had like minus 2 as the maximum value here, then instead of putting minus 2 there, we will put a 0 here. So that way, what will allow it will allow us to do is that we can find sort of not the entire sequence alignment or the global alignment, but these particular stretches of sequences that could be aligning in the middle, that are present in the middle of the sequences. So we could align those optimally, basically. So this is sort of the example of one Smith-Waterman matrix. So what it does is that it aligns two sequences, but not in a global way as we talked about in a local way, right? So in each of these paths, if we get a negative value, we just put 0 in here. And throughout this matrix, we will see that it is not the the bottom right value that is the optimal score, as in the case of Needleman-Wunsch. It is not the case, but in this case, it will be whichever value that is the maximum positive throughout the entire matrix. So basically, at any cell, if we get the maximum value, so that will be counted as the optimum alignment score. And the traceback will again, so you have to sort of follow the paths until you hit, for example, 0. So that will be here. So if you trace back, for example, in this way, okay. So these 4 sequences, so this GTGA and then this GTGA, so those will be aligned together. So as you see, we are finding like a state of subsequences that match in both of these sequences, sequence 1, sequence 2. So the alignment for local alignments can be calculated in that. So starting from the maximum score of these cells, so just have to trace back the path, and then until you hit 0, that will kind of stop the alignment there, right? So until you hit 0, you have to trace back, and then you can see which characters those grids correspond to. That's it. So this will be sort of the alignment. So ACC GTGA versus GTGA ATA. So the GTGA portion will be aligned together.
Here, consider a hypothetical example, hypothetical case. Supposedly, the 3 after 4, the path you have traced, na? So after 4, 3. Supposedly here, instead of 3, if it would have been 0, so what would be the path?
But if it had been 0, let's say in this position, right? If it had been 0, then you have to recalculate the values for these grids, right? Whichever is, let's say, adjacent to it. And then again, from whichever grid that you are obtaining the maximum value calculation, so you have to just trace back that path. So
supposedly other values are same. So in that case, the path would be 4, 2, 1.
Yeah.
And then 2 and then 1.
But if the other values are same also, then you have to sort of go in that direction, whichever is giving you the maximum, the best result, essentially. So if it is 0, let's say, so if you have to do like for this A versus A match, you have to do 0 plus 1 equals to 1, right? And then from the 2, you are also getting 2 minus 1, 2 minus 1. So all of them are giving you 1, essentially. So it should not be the case, because if it was 0, then the adjacent cells would be different also. So what you are describing in that hypothetical case is that all of the values for this trace back from 4, or for this particular grid, so that will be 1, but that will again change the entire thing, because so let's say if it was 0 and then you had 0 minus 0 plus 1, a max score of 1, right? And then the other ones are also giving you 1. So in that case, this grid is not the highest value grid, right? So in that case, you have to find out, for example, 3 was here, right? So in that case, you have to start from 3 here, I guess, because if it was 0, then 3 will be the most valued grid, or the highest value grid. So that will sort of change the entire direction of the alignment again. Hopefully that made sense.
Yeah, maybe I'll try different examples, then maybe I'll understand better. And
Okay, okay.
you mean to say ki, the path in this algorithm, the path will usually be diagonal only.
Yeah,
Mostly it will be diagonal, not
mostly yes, yes.
left.
Yeah, because diagonals will are counted as matches or mismatches. So those are basically the most probable, you can say. The gaps are much less probable, so they are highly penalized. So you'll mostly see from the alignment procedures that indels are not present there, I guess, mostly for most cases. But you have to start from the maximum value here. So from the maximum value, trace back until you get 0. That's the concept. So I guess for the next class, again, we will cover some of the other conservation related concepts and so on. So anyways, so today's session we will just stop here. So again, thank you everyone for joining. Please try to, if you have time, then you can try to join the other sessions as well, which will be occurring on Thursdays and Mondays, right? Okay. So thank you again, and again we'll meet on the next Friday. Thank you.